See, they're still joining. Okay, if anyone's just joining us, we'll start in a couple of minutes. But if you have your video audio on, you might as well turn them off. We can let you ask questions at the end using audio.
Still joining.
Time being,
They're still joining.
A one-way system for people to just listen to our speaker. I'd say we'll start about 60 seconds. I don't see the numbers going up, so any moment now I'll give you the okay to start. Not yet. Any second though. Let's get a bigger screen. Actually, I'll have one the next time, won't I? Those new laptops or PCs. Okay, it's 7:05. One more person just joined us. Well, good evening, ladies and gentlemen, and welcome to this monthly lecture of Astronomy Ireland. Tonight, we're delighted to have James Fradgley, who's going to talk about life starting on the Earth. That's our main event of the evening. I'm David Moore, the chairman of the club, also the editor of our magazine, Astronomy Ireland. And just to give any people that are calling in late a chance to not interrupt the main event of the evening, the talk, we'll just tell you a little bit about what Astronomy Ireland does and a few events that have happened in the past since our last meeting and a few after the main talk that are coming up, including the aurora that we hope to see tonight and the big moon watch that we're running on April the 8th. So we've got a bit of past, present and future for the evening. That should take us until sometime after 8 o'clock, 8:30 thereabouts. So let me tell you a little bit about Astronomy Ireland. We've been running since 1990, so over 30 years running now, and we've become the world's most popular astronomy society. During lockdown, our membership actually exploded. We nearly doubled our membership as people turned back to their hobbies, didn't have much else to do, and they rediscovered the night sky, where if you could only travel a few kilometers physically, you could travel trillions by looking up at the night sky. So it was a tremendous boost for Astronomy Ireland. We've started hiring more staff, trying to implement some of our long-term goals over the last few months, and you're going to hear a lot more about that in the future. So it's a very exciting society to be a part of. Some of you are already members, but if you're not, we'd encourage you to join Astronomy Ireland and to tell all your friends to do so as well. We have members all throughout the 32 counties. In fact, most of them don't live in County Dublin, even though a lot of the events used to be there. Now they're all online. Everybody can participate, and we hope that they will. So what do? Well, we produce Astronomy Ireland magazine, which comes out every month, full colour magazine, the only full colour monthly magazine in Ireland. And you can get that for a few euros online at our website, astronomy.ie, or you can sign up and get it posted to you every month for a little bit less. We have a weekly email that goes out to only members, telling them what to see in the sky, and also other events that are coming up around the country. We're about to restart our Friends of Astronomy Ireland mailing list, which is just for events, and that will tell you about talks like this, other talks as well, and should be sent out roughly once a week. We're in the middle of setting that up. Should be ready to go, certainly this month. And that's been offline for a good few years, but as I said, all the lockdowns and funds it's given us has enabled us to restart that. So we're looking forward to telling 25,000 people, I think it used to be, every week what events are coming up in Ireland. We, of course, have these lectures that happen every month. If you miss them, you can get them online, but you can also actually get them on DVD. In fact, if you look back at our website, astronomy.ie, you'll see past lectures by all kinds of people like the BBC's rock star astronomer, Professor Brian Cox, the Astronomer Royal of Scotland, talks on space, telescopes, black holes, cosmology, and all points in between. So do enjoy browsing back through those. We also run our evening classes for beginners. They're running at the moment, but you can still join in. I think we're on the 4th or 5th class of the 8. They happen every Wednesday evening at the moment, but they'll be repeated in October. If you sign up now, each class is self-contained, and you can repeat the entire set of 8 classes if you wish in October free of charge. So it's actually a good idea to sign up for them at the moment. Anyway, don't wait till October. As they run twice a year. We also have watches where we set up telescopes to show people bright objects, this moon and planets, the odd eclipse, for instance. Our next big watch is actually on April the 8th, when we'll be setting really powerful telescopes to look at the moon. We do this every year around the vernal equinox, when spring officially begins in the Northern Hemisphere. So you'll see that advertised on our website. But if you want to mark your calendar now, it's Friday evening, April the 8th at 8 o'clock. The big event we run every year, though, with telescopes is up in the Wicklow Mountains, where we set up some of the biggest telescopes in Ireland, have a professionally catered barbecue, 3 or 4 top-notch speakers as well, lots of trade stands and exhibits, and make a complete evening of it. It's by far the biggest star party in Ireland every year, with up to 1,000 people coming along. It takes place in the dark skies of Wicklow, so these telescopes really do perform superbly. The views are absolutely incredible, and we're inviting everybody to come along on Saturday, August 27th. You can book your tickets now. We haven't finalized the speaker lineup just yet, but watch this space, as they say. That's something we're really proud of. We also do talks in schools, corporate talks, even talks in theatres around the country. They'll be kicking off again later this year, now that theatres are all opening up. And our talks in schools are aimed at inspiring the future, trying to get young kids interested in space. It is, after all, the future of the human race. We also have a huge media presence, doing about 500 media interviews a year. That's what keeps the society one of the world's most popular, by letting the general public know, that we really want them to join Astronomy Ireland, even if they have no knowledge, but have an interest in space. So please tell your friends that. There's also very active social media, Twitter, Facebook, and Instagram. I'll tell you about those in a second, because they were very useful over the weekend. And our message is join Astronomy Ireland if you're not already a member. So these meetings take place usually on the second Monday of every month, all year round. Our last meeting in February was given by David Grennan about his Raheny Observatory, an observatory he built in the suburbs of Dublin, and has been able to find supernovae exploding in other galaxies and asteroids. And he told us all about how he did that. And as I said, if you missed that talk, you're interested, you can still get it online, and you can also get it on DVD. Just go to astronomy.ie and look up the lecture DVDs there. Then we had a huge event just yesterday. We, on our social media, predict when we think there's going to be a display of the Northern Lights, the Aurora Borealis. And we predicted a display last night, and sure enough, it materialized. It was one of the best in maybe nearly a decade. I got some great pictures of it myself. Hopefully you all read the social media posts. If not, shame on you. Sign up for Twitter, Facebook, Instagram, and make sure you get the alerts. We're hoping it might last this evening. The chance it could surge again. But last night was certainly spectacular, and you'll see my photographs in the next issue of Astronomy Ireland magazine. I'm sure plenty of other people got some snaps, because for once the weather cleared up and we got to see one of these displays of the Northern Lights. We didn't just have to live on the views that people around the world with better weather than Ireland got. That was a fantastic thing to see. The International Space Station was in morning skies, if you like, getting up the crack of dawn every night for about 2 weeks, ending last Friday. But it's coming back to evening skies from this Friday. I'll tell you about that after our main speaker. Venus, the planet, has been dominating morning skies. It's a huge crescent. You've got a telescope, and still well placed on view. It's really the only bright planet that's visible in the skies at the moment from Ireland. As I said, we do lots of radio interviews. In fact, there was one done last Thursday night about what to see in March skies. If you want to listen to that, go to astronomy.ie/audio, and you'll be able to see the MP3 there. As I said, our evening classes are running at the moment, so we've been keeping very busy, as always, at running the world's most popular astronomy club. But one of the main things we do every month is invite an expert in their field to come along and tell us in simple layman's language about their field of study. We're going to take questions at the end. You can put them in the chat box, or we'll invite you to unmute your speaker at the end and ask your question in person. As said, we're delighted that we have James Fradgley, who's studied physics and astronomy, but he describes himself more of a theoretician than a practical observational astronomer. He gives many talks, mostly to astronomy societies around the world now on Zoom. He's based in Bournemouth, where he is at the moment. We're hoping his internet connection will hold up. He's given talks to the society in the past, but he's always had an interest in biology and genetics, and was a field researcher for the peppered moth study under Kettlewell. But this evening, he's going to tell us all about the fascinating topic and what we know about it, of how life got started on the Earth. Because if we know how it started here, it might give us clues to where life might exist elsewhere in the universe. So please give a warm welcome to James Fradgley. Over to you, James.
Thank you very much, David. I think I'll stop my video because my upload speed is so awful. So I'll just keep the sound going. Thank you very much for inviting me back. This talk overlaps a little bit with the talk I gave before to you. So there's a few minutes of common ground there, which I apologize for. But I do go into it in a little bit more detail this time. The thing is that there's a lot of biochemistry, and I'm not a biochemist, so don't ask me any hard questions, please. That might be difficult. Right, I will now try and share screen, and off we go. I hope.
Yeah, we can see that.
Right, you should now have that on full screen view. Okay. Have you got that all right, David?
Not just yet. I think it takes a while to upload the first.
Got full screen view, okay?
Yeah, now you're full screen.
Okay, I'll wait till you have. Okay, fine. Yes, it's actually, as a matter of fact, the answer to this question is we haven't got a clue. Well, we have a clue, but we don't know. So that's probably it, really. I've done my talk. Well, anyway, but what we do have is a number of clues. So I'll look at some of the factors there and hopefully give a feeling for some of the things that may be relevant, if you like. The idea of a primordial soup is pretty much rejected these days. It's not going to be like this slide says. Fred Hoyle, I can't remember what date this was. The likelihood of the formation of life is 1 to a number with 40,000 noughts after it. I think it's a bit sad, really, when someone who's a superb astrophysicist starts pondering about things that basically he knows nothing about. And I'm afraid we see that a lot, but we shall see by the end of this whether we would agree with that improbability. So what will we look at? We'll look at what life is, how it might have been able to start by looking at simple molecules and working up a little bit, and looking then the other way around, looking at what we have today and going back down towards simpler forms. We'll then do a bit of astronomical stuff. What about crossing space? Are we Martians? But I have to say, given the uncertainties of this whole subject, you have to consider that everything that is said may well be wrong, or at least treat with caution, if I can put it that way. So if I sound very certain, that's because that's the way I am, and it doesn't mean it's true. Right. Okay, what is life? Oh, there's a definition there. Bounded physical systems. They've got to be able to replicate, undergo evolution basically, and decrease local entropy, i.e. increasing order within their own local framework. That of course needs energy elsewhere from elsewhere. It has to have metabolism, obviously, I think. It's got to be a rather complex structure. Probably it has to be cellular. Viruses, there's an argument about whether viruses are alive. I'll come back to that in a minute. It's got to be able to replicate and evolve. And essentially, life is using its structure to make things. So it's sort of purposive. And the information is all in this genetic code and so on. So what about a virus? They go into cells and use the machinery there to do their thing. So most scientists draw a line. But I have to say that the idea that life is a thing which is independently living, there are very little things we aren't, that's for sure. So perhaps we're not alive. I mean, it's a silly argument. It's this business of our need to put everything in boxes. But viruses are a clue as to how life may have arisen. Well, what about the genome? Awful lot of research in DNA. It was thought to be the thing. If you figured out what the DNA did, you would for sure be able to understand everything that was going on. But it turns out that's not the case. It's just, well, the tip of the iceberg, I say. There's an awful lot of stuff that's not just in the DNA. DNA is information content, or indeed it's more like computer hardware, point to the way life may have started. As I said before, life is about increasing order, and to do that you need energy. And people very seldom talk about the energy. And Jeff Hester, who's an American physicist, astronomer I was talking to, he had this nice quote: "It's always the energy, stupid, in any physics thing." And I think that's true of the source. And Nick Lane, who's a very well-known biochemist on this subject of the source of life, he is not exactly rude about DNA, but says that DNA has really been taken too seriously. And the DNA Brigade thought that life was just about copying what's on DNA. It's also true that it's probably too complex to be the first replicator, and so people came to an RNA world, which we will look at. But none of this thinking takes energy into account, which is a pretty key requirement. And we'll come back to some of the details a bit later. So what we need is the energy, and we need reactive carbon, basically. We want a high concentration of the materials we're going to use. Need to remove waste, which implies some channeling or compartments. You don't want to have enzymes or catalysts that are too efficient, or it just makes a lot of one thing. If you can have such a situation, that reduces the amount of carbon and energy required. If you can imagine doing it in a little tiny volume, it's a lot easier to get stuff together than if it's the whole ocean, for example. And of course, it makes relevant reactions to going towards life more likely. So where could life have developed? Reducing atmosphere to start with. a lot of carbon dioxide in the atmosphere, and that's methane, water, and ammonia. E.C. Bailey did experiments that were actually looking for sugars, and they did this flask you see here, or heating a bit of water, circulating it around, putting a spark through it, having an atmosphere that seemed to correspond to the early Earth's, and the spark was, if you like lightning, and then it precipitates out any compounds that might form. And they found a lot of amino acids formed. And in fact, I did this experiment at school, and within 5 minutes, we were making amino acids. So they're very easy to make. Actually, Miller and Urey's experiment has been widely condemned because there was probably a lot more carbon dioxide than they allowed in their system, and that would blanket some of these reactions, but there's certainly some truth in it. But the Earth is sort of astronomical, if you like. The early sun would have been a lot more erratic in its output, and that could easily have stripped off Earth's atmosphere once it had acquired one. And it may well be that the formation of the moon enabled there to be a much bigger magnetosphere that could have protected the atmosphere such as it was at the time. But really, it's guesswork. Similarly, the question of water, all this water we have, is that normal? If you look at the ratio between deuterium and hydrogen, deuterium is a heavy hydrogen with a neutron as well as a proton. What we see in the oceans more or less matches some of the asteroids, and it doesn't very well match many of the comets. Those Jupiter family comets that do match, they have had to come from elsewhere as well. So some of them are icy asteroids, if you like. So it looks like asteroids are a good source. Deeper in the Earth, it's a different thing. But maybe our water came from stuff crashing into the Earth a little bit later. But actually, it's even messier than that. The example of Itokawa's sample suggests that some of the stony asteroids make a better candidate than just any old asteroid, and the process that produced this with solar wind is producing quite a lot of water, and that maybe is a good place to look. So where's a good place? We've got an ocean, we've got an atmosphere. I say the primordial soup, it doesn't concentrate things enough. Darwin thought about a little pond somewhere with solar heating and gas exchange, but you're not concentrating things enough. The crust was not very hot to start with, and it was probably an ocean world, no mountains or anything. So these Hadean pictures you get of volcanoes everywhere and molten crust, most unlikely. The radiation would cool the crust very quickly indeed, which is why zircons have been found that are so old. It's been suggested perhaps ice could be a template. Clays is a very popular idea because you've got a lot of minerals there together, and could that be a template that brings things together? And for the same reason, impact craters, you've got a lot of minerals coming up there. Deeper in the crust has been suggested, but is widely ignored these days. And basically the deeps is the best bet now. Black smoke or the black smokers are what were discovered first, and people thought rising sulfur rather than oxygen, and they think, aha, you know, perhaps this is how things might have started. It's certainly producing a lot of interesting stuff, and amino acids are amongst that, but it's too hot inside the vent for life, and the more complex compounds will break down. So it's not really such a good place. And this metabolizing sulfur thing turns out to be a red herring. In fact, the reaction they're using is reacting hydrogen sulfide with oxygen. So there has to be oxygen in the oceans, but there wasn't. So it's not really very good reasoning. So amino acids are okay. Lots of ferrosanes, they're quite handy. Ferrous sulfide is a good catalyst for a lot of reactions. Alkaline vents, I'll go into a little bit more detail. They have lots of these micropores where things could happen. There's a very high voltage gradient between the alkaline rock and the water inside the rock and the acidic ocean. The temperature looks pretty happy too. So what we've got is a rock called olivine, and that reacts with water to make serpentinite. Olivine is a very, very common mineral. We've found a lot of it around the universe, and all it is magnesium or iron silicate. So olivine is everywhere, so is water, so is carbon dioxide. So that looks pretty hopeful. Olivine reacts with water to produce serpentinite and hydrogen, and that makes the rock swell and bubble and makes these serpentinite. There are a lot of reactions, but the bottom one, as you can probably see there, is making a serpentinite and in producing hydrogen. And that's one of the things we want. So we've got these alkaline fluids, and we've got all the minerals we want. And what we want to do is to be able to fix carbon. We've got to reduce CO2, which is a very tough job. We've got these high electrical gradients, which, as it happens, more or less match the potentials across the membranes in our cells, which is a good guess as to why that's so, because that's the sort of pH gradient we got in alkaline vents. So we have the same thing with living cells, acidic outside, basically alkaline inside. And so it's a good guess that that's why our cells work the way they do, worked before we had cells. Alkaline vents last a fair old time, not forever. Continuous production of pores. Organic molecules are very much concentrated. So that thermophoresis concentrates larger molecules. At high temperatures, molecules are more mobile. At lower temperatures, they can get stuck. If you put things in your tumble dryer, if they get into the pillowcase or duvet cover, they can't get out again. It's like that. And larger things are held better than small ones. So we need concentration, which is important. We can also precipitate fatty acids to make them into potential membranes. And it's also possible that we could get polymerization going. And we'll have a look at some of these things, but not too much. But first, what about getting usable carbon? Reduction of CO2 is very difficult. We want this reaction of getting the carbon and the hydrogen to react together to produce formaldehyde and water, but really what we always find is it's going the other way. And in the lab, it's extremely difficult to do. In order for it to work, the carbon dioxide must be at a much lower pH, that's in i.e. more acidic. If the hydrogen, say, is at pH 10, that's very alkaline in the vent, then the hydrogen readily gives an electron. If the carbon dioxide is slightly acidic, it's carbonic acid really, but it readily accepts an electron, and so in with a chance of making it work. And the mechanics essentially are as simple as this. To form complex or more complex organic compounds, we can do that from scratch, from just using hydrogen and CO2. And so that's a good idea for how stuff may have happened, maybe. I mentioned the proton gradient before. It facilitates, as we see, the making of compounds, and then in the little pores they get concentrated. So you've got a sort of thing like a battery, really. I mentioned the cell wall voltage, and if we started off with this free lunch, if you like, then developed a lot of machinery that uses it, and that's what we do now. So it all sort of somehow seems to fit. This is a bit of a fussy slide, and it's totally hypothetical. If you start off with the alkaline vent and acid seawater, proton gradient, so you've got a little bit of protein doing something there. In the middle one, you've got another thing forming to pump stuff in and out. And then on the right, you've got a cell wall that doesn't leak, and it can now pump things in and out as it wishes, and it can be independent. In fact, the red bit, membranes, actually are very easy to make. So it's not really a problem. The more difficult bit is getting these proteins to work in such a way that you get from the first picture to the third picture. Just a little chemical aside. Diamide phosphate, DIP, is catalyst. It helps things assemble into much longer chains, and it will add phosphate groups to bits of RNA and DNA, which is key to their structure. It can also form lipids, trying to make cell walls. It can also combine amino acids. Lots of amino acids is basically making short proteins, if you like. And it's likely that these rather small compounds were widespread in the early oceans. So we have to have both of these, really, with imidazole and DAP, but they're both likely to be there with the catalyst imidazole. Well, that's one theory, as I say. We can see the components being made fairly easily, I think. Well, we need replicators, enzymes, membranes, metabolism. We need the whole lot. We can make together, and that's the stumbling block, really. We haven't got that to work yet. And some people are saying, well, perhaps just making the components and hoping they'll do something is not the way forward. We need to do something different, as say the everything first hypothesis. But if that's so, maybe there are other places you could do it where you've got all the components together. It also is a bit of a problem if you have to have everything first, because you've got significant differences between archaea and bacteria, especially in their membranes. Anyway, still very much an open academic discussion. Well, here's the tree of life. Here's us, animals. Down the bottom here, we have Luca, the last universal common ancestor. A lot of people have said, well, perhaps if it was hot springs, we need so-called extremophiles, things that like boiling water or whatever. The extremophiles we have today are highly evolved to get there. And I think you really want to have something that can evolve without having to be extreme. So I think they're a bit of a red herring myself. Luca is an interesting one because if we've got different membrane systems in bacteria and archaea, if there is a Luca, it would probably have to precede the development of these membranes, or maybe there isn't, and it happened more than once. What we're looking at, we don't know. It's lost in the mists of time. Anyway, we talked a lot about membranes. However, given that all the same biochemical pathways are used in all of life, it certainly appears to be a one thing only. So these membranes may be a later development, and maybe life started without membranes at all. We don't know. Micropore size is very small indeed, and so you can have, if you like, a cell-sized pore doing all sorts of prebiotic and hopefully becoming biotic activity. In addition, the pore walls can be so thin that they're semi-permeable, so you can clear rubbish out if you need to, acting as membranes. So it looks, the whole thing sort of sounds sort of vaguely feasible. So that's a bit of bottom-up thinking that gets you to a lot of the components of what we have here. And I'll spend a little time talking about DNA, deoxyribonucleic acid. As you see, deoxy, less oxygen, ribose is a sugar, nucleic part of the nucleus, and it's slightly acidic. DNA, the coding in DNA, it codes for proteins and a number of other things as well. The genes are basically making proteins, and proteins are central to how our bodies work. And they are also catalysts to make reactions go at a workable rate. So we have to have proteins to make DNA work, and we have to have DNA to make proteins. So we've got a very chicken and eggy sort of problem. So looking at it a little bit more detail, what we have is backbones on the outside, and then nucleotides joining together the backbone, and any nucleotide can attach to any sugar. So you've got phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. Now that's all there is to it. And then these nucleotides that connect to each other. It's really not that complex a molecule. And what we have is that they align such that the thymine can only go with adenine, and guanine can only go with cytosine. And I'll call them A and T and C and G because it's a lot easier. So that's basically what we have, and it's these that give us the codes that we were looking for. So each of these pairs is the same size, so that's fine. They're joined by hydrogen bonds, which can easily be pulled apart, and each half can only make the opposite half, the negative of itself, if you like, the A to T and so on. And that's how the polymerase chain reaction works that we all know and love, I think. Basically, if you heat some DNA to about 100 Celsius, you split it into two separate lengths, takes about a minute, flash cool it to stop it all joining together again. You add some polymerase, that's to say enzyme or a catalyst, and the materials it needs, and in about a minute. So in about 2 or 3 minutes, you've doubled the amount of DNA you had. So repeat that 30 times. So in an hour, you've got a billion-fold amplification too, because you go 2, 4, 8, 16, and so on. And that's how DNA matching is used, and indeed a lot of tests, biological tests. Would you really want it to be the way DNA is? Some people say, well, if the strands are a bit stronger, perhaps it would be better because it's be easily separated, but natural selection wants mutations. If there was no mutations, there would be no selection, there would be no evolution. So you want some mutation, but you don't want it to be completely out of hand. Other nucleotides have been found, 4 other pairs that seem to work just as well. But it may well be that because of mutation, that was a good reason to stick with the 4 we have. We'll see a little bit more about that in a minute. So the 3 of these nucleotides, if they come in triplets called a codon, and each triplet codes for an amino acid. Well, with there are 64 possible arrangements with putting 3 of these together, but we only use 20, so there's a lot of duplication there, and 1 or 2 other functions as well. But if you see the red ones there, you'll see that the 3rd one is a dummy position, and that gives us a clue as to the past. So these triplets, I say, code for an amino acid, as you see, and chains of amino acids are what a protein is. Proteins can be enzymes, or they can be other things. However, a lot of amino acids only use 2 bases, and if you only had 2 bases, perhaps that's where we came from. That would code for 16 amino acids, and perhaps the earliest life did manage with that. That really looks a pretty likely case, looking at the way these things are structured. There's another slight twist in the whole thing with RNA, which I'll look at in a minute compared to DNA. But RNA tends to use uracil, and DNA uses thymine, and the difference is there's a methyl group, CH3, attached to the thymine ring, whereas it's just hydrogen with the uracil. And the bonding site's the same, so that's no problem. Either of them can bond with adenine. However, there are some thoughts. You need energy to add the methyl group, so you'd think that uracil was better. And T can only bond with A. However, U can cause errors, as you will see in a minute. It becomes uracil, and then it wouldn't be combining with guanine. It would be combining with adenine. You know, so you've changed. You've had a mutation there. There are repair mechanisms in DNA to try and get rid of uracil because it causes too much mutation. And also uracil can orient in different ways, which means it can bond in different ways. So you really don't want uracil, which is what we have with RNA, but you don't want if you want a more stable structure. The other difference is this ribose, the sugar part of the backbone, and the DNA is more stable because the strands are better attracted to each other than in water. But we'll see a little bit about, yeah. So I'll have a look at some of the types of RNA. There are hundreds, probably thousands of types of RNA. We'll look at messenger RNA and transfer RNA because it explains some of the process that's going on there, which maybe is old hat for everybody. We've certainly known about it for a long time. What messenger RNA is the DNA unzips a bit, and then it makes a copy of itself, not with DNA, but with RNA, because you've still got the nucleotides separating, and off it goes. You've got a strip of, if you like, a copy of the DNA. The transfer RNA is a slightly more complicated animal, and there's 64 types of transfer RNA, because one end has the triplet, look at the codon, and the other end has an amino acid, always the same amino acid for each got. This triplet, it's this amino acid, which was the big table we saw a minute ago, and it's actually in a bit of RNA. So if you like, the DNA is like hardware. It doesn't say what's to happen. The transfer RNA is saying, oh, I know what's to happen because it's the thing at the other end of my molecule. And amino acids are the output from all that. What happens then is the mRNA goes into a thing called a ribosome, which are quite complicated animals. So it's a good question how they arose in the first place. mRNA is just read through the messenger RNA, so that's reading, if you like, the stuff that's been taken off the DNA, and the matching amino acids come from the transfer RNA, and they're stitched together in the right order. And then you've got your protein. The transfer RNA can pick up another amino acid, and off you go again. And that's really how these RNAs work. But ribosomes have ribosomal RNA and a lot of proteins in them. It's quite a complicated animal. So how do we think that might have started? Well, if you had two compounds, A and B, coming together to make C, if you've got a catalyst, you can speed the whole thing up. Well, supposing C is the quicker, you get a runaway chain reaction, and that has been done in the lab using RNA as the catalyst to make itself, self-catalysis. And some of the ribosomal RNA molecules do indeed act in that way. And so it seems like there's a number of clues as to the idea that perhaps before DNA was there, there was an RNA world. So autocatalysis or cross-catalysis, you'll get an awful lot of stuff, basically. Can't be very big, because RNA is not that stable. A lot of viruses are based on RNA, not DNA. COVID's an example, flu's another. Explains that why these things mutate so rapidly, because they're not that stable. And if it's too big, you get an error catastrophe and the whole thing doesn't work. So it's only really good for smaller molecules that contain less information, if you like. So RNA would be a small molecule basis of life, if you like. So do we need a template? Well, we are reaching down to not far from where we started. We've got to join lots of small molecules, serpentinite template possible, something in the pores. A lot of simulations have been done, but they haven't answered the question. Obviously, we need to get from RNA to DNA, and we don't know how that would have happened. But once it's happened, we're away. So there are a number of steps there we really don't know, but we're getting, if you like, a look into how something might have happened. There is this breakthrough problem, which is the fact that you need proteins and the RNA or the DNA. You have to have both of them. And as I said, the RNA enzymes in ribosomes is, and indeed the fact that there are a lot of RNA viruses is a clue that perhaps a lot of stuff started that way before it had cells to work on, that it was doing some of its own stuff perhaps. Well, as we know, life appeared on Earth very quickly indeed after the late heavy bombardment, a bit under 4 billion years ago. So either it's jolly easy to do this, or the panspermia argument is right. If it's panspermia, that's sort of rather begging the question, I think, but we'll have a look at that. There are a lot of organic molecules in space. Fairly recently, they found that 67P, Churyumov-Gerasimenko, they found amino acid glycine, the simplest amino acid, and it's they got very excited. It's a precursor for life. No, it's not. It's just a small molecule. It's true that life uses it, but that's a separate point. But there are an awful lot of amino acids out there in space, a lot of ethanol too, so that sounds nice. Sugars, polycyclic aromatic hydrocarbons, even buckyballs, carbon-60 molecules. So we've got an awful lot of stuff out there in giant molecular clouds. And even in the solar system, you look at the orange color on Titan or Pluto's surface, it's taking all these things, nitrogen, ammonia, methane, carbon dioxide, water, and solar radiation is breaking things up and causing reactions to happen, such that you get this sort of brown gunge. We know on the surface of comets and dust grains, they're very dark because they've got a lot of this sort of stuff on them. So there's an awful lot of chemistry going on, but there's a lot of radiation out there. So complex cells, complex molecules, I mean, are most unlikely to exist. But a lot of foodstuffs, if you like, for life is out there, that's for sure, which would have been raining on the Earth. But maybe we don't need that, but nice if it's happening. So what about crossing space? What about this panspermia idea? The Surveyor 3 probe landed in April 67, and Apollo 12 retrieved the camera, and there was found to be some Streptococcus mitis inside the camera still alive. Now, Streptococcus mitis is a bug that's up inside all our noses, and so it's quite possible that someone breathed on it or something happened and that there was contamination. So we really don't know, but a camera would be a pretty good shield for things to survive. Radiation is the big problem, basically. It's a killer, whether it's just the UV or flares or cosmic rays, but it's not a happy thing for life. So it's a question of how good can spores be? If you have a cell that can become a spore, it's basically genetic material with a hard shell around it, so-called cryptobiotic. It's just not really alive until it gets something to get it going again. And some work has been done to see how well spores can survive. Well, we don't know Streptococcus mitis. We've looked at an experiment was done, gosh, it's nearly 40 years ago in a satellite where there was a high survival rate, but it wasn't outside it wasn't too far from the Earth, a bit shielded anyway by our magnetosphere. Other tests have shown much lower survivals. Things can survive a very long time. Viable spores have been found in geologic deposits that are very old. So things can survive really for an amazing length of time. And some some bugs can do amazing things, but they're highly evolved to do so. But with the extensive repair mechanisms, this stuff is Deinococcus radiodurans is quite happy to live inside nuclear power stations and places because it's got extensive repair mechanisms. So there's a lot of thinking about how things might be. I couldn't find a quantitative answer to this one, but some of the spores survived for 18 months when exposed. So it doesn't look like long survival can happen if you're exposed to space. People have proposed, well, it'll be inside things, perhaps inside a dust grain or something for interplanetary travel. And for interstellar, where it needs a meter or more, perhaps, of shielding, if it's going to take millions of years, then perhaps inside comets or something. So those have been suggested. Well, there you go. You may remember, however, it got terribly excited about the Allen Hills 8401 with that little thing that looks like a caterpillar, but that has been sort of roundly rejected now, I should say, and as it was a Martian meteorite. If you had a largish body impacting Mars, a couple of things, stuff that's blasted into space does not get very hot. And when it lands on Earth, it doesn't get very hot either. You think smashing through the atmosphere at high speed, yes, you get this sort of enormous meteorite streak through the sky. But inside it, it's not hot. And indeed, if you ever came across a meteorite that just landed, it's still got the cold from the depths of space in it. So if there was life on Mars, it could well have made it to Earth, is what it boils down to. There are other places in the solar system that have been suggested for life. Enceladus, with its jets coming out of the South Polar region, Titan, with a completely different sort of biochemistry based on methane and ethane lakes, and possibly Europa as well. So those are obviously places we're going to look. The likelihood of life distributing itself in an interplanetary sense is going to be very directional. And so it'll fall towards the sun more easily than away. So it's possible that some stuff from somewhere has fallen on the Earth. So here's the conclusion. Life started very early on Earth. I would argue that we're starting to see that it's probably easy to do so, rather than that it's come here from space. I suggest that biochemistry is now a lot more advanced than when Fred Hoyle made his statement, and so he hasn't scored a goal there. The alkaline hydrothermal vents look really very much like this thing. There's so much going for them. And the sort of structures that provide alkaline vents are likely to be common. It's quite likely that what's coming out of Enceladus is coming, that there are such things in there. We know that there's some, the trouble is the Cassini probe was unable to say what material was coming out, but there were things other than just the water. So we need to go and check. And yes, we may indeed all be Martians, but that doesn't solve the source of life, because it's probably easier to do here than to be Martians. But if our DNA is the same and we find life on Mars, then that may well say something about it. None of that's anything to do with complex life. We're just talking about the very, very simplest life. So there we go. finish it. I see there's a couple of chats there. Things.
Are you happy to take questions now, James? No?
Yeah, I see that my internet connection is fluctuating. I'm afraid I've got a crap internet connection. It's true. I'll take questions. Whether I can answer them or not, it's a moot point.
Do we have any in the chat then? Any questions in the chat? I didn't think I saw any.
No.
Click the right thing. Well, we'd invite people to unmute their microphone and ask James directly. If you're on a laptop, you can just press the space bar. As long as you hold it down, you're unmuted. Handy way of doing it. Or just unmute, ask your question, and remute. So while you're all thinking about your questions, I might ask one myself for going back to viruses. So a virus needs to take over an actual cell to reproduce itself. So there couldn't, if RNA did precede DNA, there couldn't really have been any viruses before the DNA, could there?
Well, it's quite possible that if there was an RNA world, that you would have things like viruses that had more capability than today's. And they've lost that capability because they don't need it, because they can use cells to do it for them. So it's quite possible that you had more capable viruses, if you like, than we have today. But as I say, it is really guesswork, because of course we've only got today to look at. So, but I would hazard a guess that that's quite possible. Got a question here from Ed Molalarki. Is a brain necessary for life, or is the brain only associated with intelligence? First question. I would say it is only associated with intelligence. The sort of life we're talking about here is bacteria, things like that. Not much of a brain there, but there is some order and process going on that we call life, and whether you would call that a thinking thing, I think not, but there is certainly a consistent structural thing going on, which implies some sort of logical coherence. Is there any overlap between life and intelligence, was the second part. Well, yes, I guess there is. There's life without intelligence and there's life with intelligence, but I don't think we yet have seen, until the Silicon Boys have done their job, any intelligence without life. When does life begin in a pregnancy? I don't know.
Dangerous question.
Yeah, I think I guess at conception, but that's just an opinion, isn't it? So where are alkaline hydrothermal vents found? Well, I know there are some in the mid-Atlantic ridge, and there are some somewhere else, but I don't know their exact locations, I'm afraid.
All those black smokers that we see, they're not alkaline.
Yeah, they're not
Are they?
alkaline, no. I think they were found in the Pacific to start with. So they're here and there all over the Earth's surface. Right.
Just shooting up there to the top, James. You see, Thomas Finnegan has a question.
Oh, okay.
Above Edward Malarkey's one.
Okay, go on.
So Tom asks, do we need panspermia to explain life, or would there have been enough complex molecules and reactions occurring, such as lightning or radiation, to have created early simple life?
Well, I don't believe in the panspermia thing myself. I think, and the reason principally is that you need to concentrate stuff. You've got to get a lot of stuff very close together and get the reactions going, which means you need something like pores in a liquid, probably water, medium. So if there is panspermia, it's got to have come from something very similar to what we have here. And if we can see that it might happen here, why do we have to look elsewhere, I suppose is where I am. The question...
Do stay with us, because we want to tell you about an RTE documentary that's coming up about life in the universe and the aurora tonight. But we have another question in chat there, I think.
Yeah, there's one from Jim. Could extraterrestrial life be very different, i.e. not based on DNA, RNA, or not carbon-based? Well, I think DNA, RNA is just a conceptual structure. And I dare say people have made suggestions for completely different, logically similar structures, but completely different chemical structures, what you might get on in Titan's lakes. Just nobody's arguing that it wouldn't be carbon, because carbon has such chemical flexibility to make complex things. So I think it would be carbon-based, but I'd guess you don't have to have DNA, RNA, but you've got to have something analogous. You've got to be able to have a genetic code of some sort.
And if there aren't any more questions in chat, I might just ask one final question.
Okay.
And that's it. You said life got started soon after the late heavy bombardment, 3.7 billion years ago. Did I read something recently about life being found, or evidence for life being found, even before that?
Yes, there's a lot of dispute about when life may have happened. And indeed, if life can arise easily, it may well have arisen and then been wiped out by bombardment. So people are always looking for older possibilities, but I don't think we've yet found anything that antedates, if indeed the late bombardment happened, because there's still some academic dispute about that. But something like it looks like it happened. I would buy that one. So I suspect you've got to have a little bit more stability on the planet before things are really likely to happen.
I see there's a question there from Natasha. Can you read that one? Could there be different biomarkers then?
Is this the sort of looking at exoplanets question?
Yes. So just leading on from Jim's question, could there be different biomarkers then we should be looking out for?
Yes, absolutely, there could. And what you have to look for is something that doesn't make sense, something that's unstable. You have to look for stuff in an atmosphere that couldn't be there, like methane in an oxygen-rich atmosphere, for example. And that's why people get excited finding methane on Mars or whatever, because if we find things that don't quite make sense, and there are a whole huge number of things for which that might be the case, yes, you've got to look for things that
stand out,
just don't make sense.
Yep. Cool, thank you.
Okay.
Well, I don't see any more questions there, so maybe we'll, can you stay with us, James, for a few minutes?
Okay.
Yeah. Well, what we might do is tell people about what to see in the sky and a couple of events coming up. And if anyone has any questions, throw them into chat. And we still have James with us for a short while anyway. And this won't take long, because I did want to tell you a little bit about that RTE life in the universe. Turn on the video again. That helps. See me anyway. But we've recorded 3 one-hour episodes of a new documentary series being made. So I know it's going down RTE Digital. It may be later this month. We're waiting to hear, certainly in the very short term. And the producer has been around the world. Well, actually, he wasn't. He was doing it all during lockdown and managed to zoom into lots of people around the world, some of the world-class experts on this subject. So it sounds like a fantastic documentary. He was very kind enough to interview me as well, to sort of tie things together with general questions and then get the real answer from the experts. So that should be a bit of fun. That'll obviously be on our social media and mailing us when we find out the exact transmission date. Until last week, but he hadn't got it as of yet. So that should be a great bit of fun and ties in nicely with this evening's talk. So let me briefly tell you what's happening in the sky. Of course, all this is in the magazine, except for the aurora. So the aurora was seen last night. We did predict it on our social media. In fact, we predicted the day before. Saturday, Friday, I think we said it was going to happen on Sunday. Saturday, we repeated that, and it materialized. It actually happened earlier, but then it surged again. So we thought maybe only the continental US was going to see it, but then there was a bit of a lull, and then it surged again, which meant Europeans got to see it, including yours truly. It wasn't great with the naked eye, but it came out superbly on the cameras, as is often the case. So those pictures will be going into the magazine. Still haven't had time to process them, all the preparations for today. Other than that, I would have shown you a quick one. That's the aurora. There's a chance it could surge again tonight. Now, these predictions are very dodgy, but it looks like midnight for a couple of hours is the most likely time. That said, trying to predict aurora within hours is not easy, and I wouldn't trust that. It could be happening now. In fact, if anyone's in the rural location wants to look out, let us know if the aurora is actually happening. I haven't had time to check the satellites during our talk. But just if you've got nothing better to do, just keep watching the northern horizon from dusk till dawn. That's all we can really say. Might not materialize tonight. I saw it till about just after midnight, and then nothing happened. Waited till about 4 a.m. Nothing much was happening after that. That's the way with aurora. They're quite unpredictable, but there was a big explosion on the sun, which enabled us to predict that Sunday night was probably going to be the night. In fact, parts of it arrived early. So you can never really tell within 6 to 12 hours exactly about these aurora until they're actually happening. So that's the aurora. Watch out for it tonight. And even if it doesn't materialize, there's bound to be more. You may have heard me on RT Radio 1 with Claire Byrne talking about all the aurora alerts that we've had this year. We're not near solar maximum yet. We shouldn't be having this much solar activity. So the sun is really surprising people at the moment. It may go very quiet for the rest of the year. If we continue this trend, there'll be lots more aurora. One thing we are going to do is revamp our travel club. It went 4 times up the fjords of Norway into the Arctic Circle to watch the aurora. Once we actually flew in the Arctic Circle in Tromsø, and every time we went, we had fantastic views of the aurora. They're always much stronger up there. Even last night, we were really, I think it's actually quite high up, about halfway up the sky, which was quite good. But usually they're out over the Atlantic Ocean, north of Ireland. People on the north coast often get the best pictures, not because being that bit extra north compared to Cork is what's doing it. It's just that there's no towns or streets or even mountains or trees or buildings or anything if you're on the north coast of Ireland looking out over the ocean. They have an ideal viewpoint as long as the sky is clear. There is a bit of a moon around. In fact, a full moon later this week, and that is brightening up the sky, but it didn't stop the aurora coming out nicely on the photographs. So that's a bit about the Northern Lights. Hopefully we'll all have some luck this evening. Back to the regular programming. Well, planets on view is only really Venus. Certainly only naked eye planet on view, but it's very spectacular in morning skies, rising 2 hours before the sun. So even just before sunrise, half hour before sunrise, you'll see Venus blazing over in the southeast. So do look out for that. If you've got a telescope, its disk is quite large, 25 arc seconds across. More on that in the magazine. And in fact, Venus is near Mars. Now, I thought this would be easier to see, but I managed to spot Mars. It's about 5 degrees below Venus. In fact, on the 16th Wednesday, technically they're closest, just about 4 degrees below. But for several weeks, Mars is going to be close to Venus. You will need binoculars, though, because Mars is 200 times dimmer than Venus. Venus is actually on the near side of the sun to us, and Mars is on the far side of the sun. It'll be a fantastic sight, Mars will, at Christmas in December or so. But at the moment, it's quite dim. But if you've got a pair of binoculars, do check out, and you're looking at Venus, do check to have a look for Mars just below. There's a diagram in the magazine. In fact, Venus has had its maximum separation from the sun this week. So it's a great time to be watching it, 47 degrees away from the sun. At the end of the month, just skipping ahead, on Sunday the 27th, Monday the 28th, these are mornings, remember, they're all in the morning sky. Mars, Venus, and the moon all get together, and some lovely diagrams in the magazine. That's a Sunday morning, so it's not a school day the next day. So do plan on getting up early or staying up late that particular night and use the diagrams in the magazine. Other things are happening. I said there was a full moon coming. Technically, it's Friday morning, so Thursday night the moon will look full. Friday night will look full. It's called the worm moon, this particular full moon. Not really important what its name is, but it will brighten up the sky. And then that same night, Friday night, the International Space Station starts its series of evening passes. Now, the first couple of days, it'll be fairly low down, probably in twilight. But after that, then we can see it till April the 3rd. We've got about 2 weeks to see it. And by next week, it will be blazing across the sky. So watch out for that. We'll issue daily predictions on our social media, Twitter, Facebook, Instagram, so you can get those for free. And if it passes near one of the planets or the moon, we predict where you need to be in Ireland to see that. So there are other ways of getting space station predictions, but stick with Astronomy Ireland, because we'll tell you where you need to be to see it pass in front of the moon. And over the decades, I've managed to see that happen a couple of times, because 1, it happened from Ireland, 2, it happened near where I lived, and 3, it was actually clear. A lot of things to come together in Ireland to actually see that. But other people have seen it as well, so watch out for all that fun from Friday on. That's more or less what's happening. Looking a bit further afield, our next lecture will be April 11th. So on April the 4th, the Pleiades star cluster is very close to the moon in the same binocular field of view. There'll be a diagram for that in the next issue of the magazine, the April issue. It'll be out at the end of March. And then Mars will actually get close to Saturn. Now, they're a fraction of a degree apart, so about half a moon diameter, much closer Mars gets to Venus. But they're both dim, and whether they'll be well seen or not, I'm not sure. But we'll put a diagram in the magazine. April the 5th is the morning to watch. They'll be reasonably close to the morning before and after as well. And the only thing that happens, and we put this on our emailing list that goes out to members only, usually every Wednesday morning, is that there can be giant sunspots, and there have been a lot of them on display on the sun. They are the things that cause the northern lights. So do watch the members only mailing list. If you're not a member, do join to get that. So a lot of the diagrams that are in the magazine, which you can buy online, astronomy.ie/magazine, and you'll see the link there. Three other events briefly coming up. Our evening classes I mentioned already are running now. You can enroll today, tomorrow, and do them every Wednesday and repeat the entire course free of charge in October. We also have our annual moon watch on April the 8th. It's a Friday night at 8 p.m. The clocks will have gone forward by then. That's just about sunset. But the moon you will see easily, very high up. And this time of year, around the vernal equinox, which is March the 20th, by the way, and that is when spring begins in the Northern Hemisphere. Don't believe all these meteorologists and other people saying, oh, it's the 1st of February, oh, it's the 1st of March. No, the equinox is an astronomical event. It's linked to the Earth's tilt. It always has been, always will be. But for some reason in Ireland, they seem to like moving the date around, but it always will be the equinox. So March the 20th, 3:30 p.m. is the exact start of spring in the Northern Hemisphere, including Ireland. You need to get that straight up, written into RT, like complaining. So that's the equinox. But as we're saying, our moon watch around this time of year, the quarter moon is very high up, and that's great for getting the clearest views with telescopes. So we always have an annual moon watch, usually just after the vernal equinox. So it's April the 8th, Friday night, 8 p.m. Details are on astronomy.ie. Bring all your friends. They will absolutely be flabbergasted by not just seeing the craters on the moon, but the mountains inside the craters on the moon. So watch out for that. The last event to tell you about is our next lecture on Monday, the 11th of April. It's gravitational wave astronomy, an update. It's going to be given by Andy Briggs, who's zooming in from Spain. Andy has given some superb talks on all aspects of cosmology in the past. And this one promises to be a humdinger, because gravitational waves, you may remember, were recently awarded, or a study of them was awarded the Nobel Prize. It's one of the big areas in astrophysics produced by giant black holes or neutron stars, or probably both, doing crazy things when they merge, the kind of explosions that make the world's entire nuclear arsenal look like a firecracker compared to the sun. So that's going to be absolutely fascinating. He's a brilliant speaker, originally from the UK, but now living in Spain. Don't miss that, Monday, the 11th of April. Get your tickets for that on astronomy.ie right now, and I would highly recommend that talk. So that's all the events that are going on. Do order the magazine if you don't already get it. Make sure you sign up for some of those social media. We can tell you about International Space Station and Aurora. And I'm going to close the meeting there, unless somebody has had any more questions for James. I don't see anything in the chat. So unless somebody, I'll give you a few seconds just to unmute your speaker and ask him.
How did it work, David, in terms of the line speed? Did it come through all right?
Most of it did. There were a few times when it broke up. I don't know too much about Zoom, but it seems to sort of buffer what you're saying. And if it knows the connection's not that good, it plays it back rather slowly and then speeds up.
That's right. That's Zoom. Yes.
Yeah, but it's still, I lost a few words, I'd say 3 or 4 times, I'm afraid.
Oh dear.
But we certainly got 99% of your talk, and absolutely fascinating, by the way. So I forgot to thank you for the talk.
Thank you.
I learned a lot. This is one area that's changing so rapidly as people discover new things, and it's nice to have a summary from an expert. Some of your diagrams showed you really do know your stuff. Explain it all to us. And if some of it went over people's heads, you don't need to know everything about astronomy. The fact that we know how these molecules, how complicated they are. I mean, one almost question I was going to ask was, this just looks so mechanized that how could the universe just fall together such that these kind of incredibly complicated molecules, I mean, what, there are 3 billion base pairs in DNA? So there's tens of billions of atoms all working together, human beings at least. And that chemistry could produce something so incredible, it almost makes you think about intelligent design. That's personal opinion, but I don't know if you have any ideas on intelligent design, or is it just chemistry?
I think it's chemistry.
Isn't it amazing though that carbon atoms can do this kind of incredible chemistry?
Well, until we've succeeded in doing something in the lab, you know, we just don't know. We have some fairly significant stumbling blocks. 10 years ago, I would have said, I think in 10 years we'll have got it sussed, but we haven't. So
yes, I know we talked about this in the RT documentary, but some of the researchers looking for intelligent life elsewhere, and therefore assuming that it can get started fairly easily, as I think you were suggesting, have said, including I think Frank Drake said this, that he expects that we will find evidence for intelligent life within our lifetime.
Yeah, well, that was my previous talk, wasn't it?
Yeah. Which, by the way, should be on the astronomy.ie website if anybody wants to see that. So I don't see, do I see something came in chat?
No.
Just a thank you. Great. Well, that's where we'll leave it. So thanks to everybody, all of our staff and volunteers making the event work well. But a big thank you to James for giving another superb talk. Go out and enjoy the aurora.
Thank you.
Good night, everybody.
Thanks very much. Bye.
Night all. Take care. Thanks a million, James.
Nice all. Thanks, James. Good night.
Great. Well, if Sonia's there, you want to end the meeting, or will I?
Thank you very much. Thank you.
Thank you. Thank you very much, James. It's a great talk. Really enjoyed it.
Thank you.
David, I don't know, can you do it there as co-host so I can sign up?
Let's see, I'll stop the recording. Here we go.
Okay.
Make sure, yes, I am. Okay, and if I click leave, most people have already left. Bye-bye, James.
All right. Bye then.
Thank you very much again. I'll talk to you tomorrow, Sonia. Yep.
Yeah, okay, thanks, David. Right,
All
I'm going to
the
end
best.
this.
Bye-bye.
Right, bye.